Lab rats are overwhelmingly male, and that's a problem. CNN, the data didn't make sense. 5 years ago, University of Maryland researcher Alisa Morse claimed was studying pulmonary hypertension, a type of high blood pressure that affects the artery in the lungs. In human cells she had cultured in her lab, but the results she was seeing just didn't stack up. We had these huge error bars, it didn't make any sense, she said, and we said, okay, let's just graph it by male versus female, and what we found was really interesting. The blood vessels in the lungs of people with pulmonary hypertension take up more glucose, and she found the female cells metabolized the glucose in a way that changed the protein that was critical to blood vessel function. In other words, the sex of cells became an important variable that affects the outcome of the research. Because we were grouping our sexes together and we were missing the differences. We were getting the average with a big deviation, said Klein, an associate professor at the University of Maryland's Fischel Department of Bioengineering and the director of the Vascular Kinetic Laboratory. I was shocked when we saw the cells themselves were different based on sex. Concern has been growing in recent years that ignoring or downplaying differences in sex as a biological variable, whether in cells under a microscope or in lab animals, is undermining biomedical research at the earliest stages. This matters because many diseases, including COVID-19, affect men and women differently, and missing sex-based differences can make misdiagnosis and mistreatment more likely. When researchers don't consider sex as a biological variable, that means we have incomplete data. And if we have incomplete data, we run the risk of making judgments that are incorrect. And incomplete data and erroneous conclusions in preclinical research ultimately can have an effect on the health of all of us, the health of women and men, said Kyron Hunter, Associate Director for Basic and Translational Research at the National Institute of Health Office of Research on Women's Health, which is holding a conference on sex as a biological variable this week. The default lab rat is male. While it's often unclear what sex the cells used in lab research are, and to what degree female cells are underrepresented, the default lab rat has long been male. One study from 2011 found that in neuroscience research, male animals are used 6 times more often than females, and the figures from a more recent analysis in 2017 suggest we have only improved marginally. Women have been routinely included in clinical trials since the 1990s, but integrating female animals into basic and preclinical research, which is key building block for studies in humans, have been much slower, said Hunter. Women are more likely to be misdiagnosed than men when it comes to a wide range of conditions, including heart attacks and ADHD. and women typically experience the more and more intense side effects from pharmacological drugs. Some drugs are more effective in men than women, including common over-the-counter ones such as ibuprofen and naproxen. Both forms of non-steroidal anti-inflammatory drugs called NSAID. And it goes both ways. Aloxicron, a drug approved to treat severe irritable bowel syndrome, is only approved for treatment in women, as it is largely ineffective in men. We are saying here that not all researchers need to study sex differences, but all researchers really should consider how biological sex can impact the questions that we are studying, Hunter said. In the case of research into COVID-19, it is not clear that sex as a biological variable is constantly being taken into account, even though men are 3 times more likely than women to be admitted to the ICU. What is being done? Major funders of scientific research, including the U.S. National Institutes of Health, which handles 80,000 grants a year, have required the research we found to take account of sex as a biological variable. Since 2016, NIH grant applications are required to include both male and female animals or cells in both the design and in analyzing the result. Hunter said that there have been signs of progress, but a 5-year report card called for greater action. The NIH doesn't have any mechanism to ensure the funded research adheres to what was set out in the grant application, and some feels the efforts made in grant applications are only lip service. It's been nearly 6 years, and we have seen the needle move in any considerable direction in terms of even understanding what the sex differences at baseline level in between males and females, said Aditi Bhargava, a professor in the Department of OB/GYN and the Center for Reproductive Sciences at the University of California, San Francisco. Bhargava, who is the author of the new scientific statement on sex as a biological variable for the Endocrine Society, and others said that high-profile scientific journals like those published by Nature and Science need to do more to ensure the papers they publish take sex as a biological variable into account. It's up to the journal and the scientists who peer review for the journals to start holding people accountable and say this doesn't deserve to be a high-profile paper because it only studies males. It can't be that impactful if we don't know the answer to the science question in females, said Rebecca Shansky, an associate professor in the Department of Psychology at the Northeastern University in Boston. Shansky wrote in Nature Neuroscience in March that considering sex as a biological variable will require a global shift in science culture. Many journals asked the authors to adhere to SAGA guidelines on sex and gender equity in research, but that doesn't mean that researchers have to comply with them. We are not currently monitoring compliance with the SAGA guidance, but We may, in the future, ask the authors to provide a clear disclosure as to whether the study adheres to Sager guidance, said Soumya Swaminathan, head of editorial policy and research integrity Nature portfolio. She said Nature does require the researchers to fill in the details reporting summary that includes the sex of lab animals. An independent study published in 2019 in the journal BMJ Open Science, which evaluated the effectiveness of these reporting summaries, found 52% of the sample of Nature journal papers published after the reporting summary was implemented reported the sex of experimental animals compared to 36% in the sample of articles on similar topics published by journals from other publishers. Not just hormones. For a long time, female lab animals were thought too hormonal and messy for science, said Shansky, and one of the main reasons given for not using female mice was that they made data more variable as a result of the female reproductive cycle. But this is a myth, she said. Hormones aren't a uniquely female problem. The testosterone levels of male mice can also vary widely. The push for greater awareness of sex as a research variable is not just about using more female lab rats. It's also important not to take the behavior of male mice as a baseline for research, Shansky said. In her lab, she has been studying fear behavior in mice, and the typical fear response in mice have long been thought to be freezing. However, she found that female mice will often exhibit an escape response. They try to escape the boxes they are afraid. They are just showing it differently. So, say, if we were looking for drugs that reduce fear, say to treat PTSD, and we were looking for a freezing response when the breathing goes down, we wouldn't be looking for the right response, she explained. If you based your interpretations in the behavior of male mice, we might misinterpret the behavior of female mice. This could throw off the result of research, she said. We are still learning about differences between the sexes in both animals and humans, but they go beyond the hormones and the reproductive system. A study published last year in the journal Science found that over 13,000 genes are expressed differently between the sexes. The researchers also identified the sex biased patterns of gene regulation that are linked to over 50 bodily traits and functions. These differences don't always matter, that shouldn't be exaggerated. For example, the popular sleep medication Zolpidem, better known as Ambien, lingers longer in the blood of women than of men, causing drowsiness, cognitive impairment, and increased traffic accidents. For these reasons, the FDA in 2013 halved the recommended dosage prescribed to women. One subsequent study into the drugs after the FDA changed its recommendations suggested that it was body weight, not sex. That was the key biological variable. However, many researchers agree it should be taken into account. Bhargava said disease should be thought of as a destination, and researchers needed to pay more attention to the journey at the biochemical and cellular level. I want to come to London. If I coming from San Francisco, I could take a non-stop flight, or I could come via Washington D.C. and Paris, she said. The outcome is that we've all reached London, but the paths are divergent. Men and women can take different paths, and they may cross and intersect to reach the same destination, or in this case, in disease. But that doesn't mean that they started off in the same way, or that every single step that they took to reach that destination was identical. Challenges. For Klein at the University of Maryland, the realization that the cells she worked on differed by sex has completely reshaped how she and her lab work. I had never thought about the sex of my cells before, she said. I just thought we take cells out of the person and we get rid of the physical and biochemical differences that happened in the blood vessels of men and women, and we should all be the same. Information on where the cells comes from isn't routinely available, and she now spends time tracking down the details. She also has twice as many samples in each experiment, which added up in terms of cost and time. Plus, it can be hard to confident about the sex effects in experiment. We have been asked how many cell donors we need to be sure that an effect is sex rather than donor related. We don't yet know the answer to that question, Klein said. But even with these challenges, she thinks the effort is crucial. People think that if you take the cells out of the living being and to put them in the dish, there is no differences anymore. A lot of thoughts about male and female differences are based around estrogen and sex hormones, but it is more fundamental than that.